Okay, so now we're going to talk about microbial disease just in general. So what do we talk about when we talk about microbial diseases? Well, we're talking about, of course, the main pathogens. And so we have viruses, which are obligate intracellular pathogens. And this is because they require our cells to make their proteins. Now we also have our bacteria, and they can be either intracellular or extracellular. Now, intracellular would be things like TB or other mycobacterium species, and extracellular would be, of course, our staph and strep. And I'll add another one for intracellular. Listeria is an example of intracellular. We also have our fungi. We have our endemic mycoses. And these will infect healthy people. And they are going to have thermal dimorphism, meaning that they are going to be a mold at room temp and a yeast at 37 degrees, which is our body temp. We also have our opportunistic fungi, and two big ones are going to be Candida, which is always yeast, and we have Aspergillus, which is always a mold inside our bodies and outside of our bodies. And of course, our parasites. And these can be simple or complex. Now, classic microbial diseases are such that you have organisms that cause a very well single defined disease. And these are things like Clostridium tetani, which causes tetanus. We have Ebola, virus, which causes hemorrhagic fever. And we have Plasmodium, which causes malaria. And then you have, of course, exogenous infections. Exogenous infections are going to be classic. It's what we really think about, that it is something from the outside infecting us. And so again, our Clostridium tetani is exogenous. It's in the dirt. And so we come in contact with Clostridium spores in the dirt and we get infected in a wound, for example. Influenza. We can never be colonized with influenza, of course. It's an exogenous infection. Neisseria, gonorrhea. Oops. We have Entamoeba histolytica. And many others are exogenous infections. So that's what we kind of think of as a classic microbial disease. Influenza virus causes flu. Measles virus causes measles. Varicella zoster virus causes chickenpox. So we have those some common microbial diseases, and these are going to be associated with pathogens such as Staph aureus that can produce many different diseases. So Staph aureus is a microflora bacterium, and it colonizes about 10% of us in general, in the general population. And Staph aureus, of all the staphylococcus, have the most virulence factors. And so if you are a healthcare provider who is a carrier of Staph aureus, you could pass on this potential pathogen to your patients that are compromised. And Staph aureus can cause endocarditis. It can be a form of pneumonia. It can form skin conditions. We'll talk about those. It has a toxin, and so it's associated with food poisoning. And so this is by a toxin. And then we also have on the other side, we have many different organisms produce the same disease. So meningitis is the example here. Meningitis can have an origin from a virus, a bacteria, a fungal, or even a parasite. So there are some pathogens that cause a specific well-defined disease, and then there are other diseases that can be caused by many different pathogens. Now, the host-pathogen interaction is very important, and it's very complex. There are, in general, 5 fundamental requirements that a pathogen has to do in order to have a successful infection. So, of course, the first, which should seem very obvious, is that the pathogen has to have some means of getting into the host. And then once it's in the host, it has to remain in a location to get established and to grow. So this could be a extracellular bacteria that grows within your tissues. It can be an intracellular bacteria that finds a cell that can support the growth of the bacterium. It can be a virus, etc. But it has to get in, it has to find out somewhere where it can replicate. In addition, it has to be able to evade their immune defenses. And so for most microbes do not have virulence factors, and they get eliminated very quickly when they enter your body. It's those pathogens that do have virulence factors that are much harder to get rid of, such as that Staph aureus we just discussed. There is going to be some sort of damage to the host. And again, this is going to depend on the type of pathogen. And the damage can be localized damage. The pathogen can go systemic, and you can get a systemic infection. So it just, again, depends. The pathogen has to have a way to be transmitted to somebody else. And again, our more contagious pathogens are our respiratory pathogens, because respiratory droplets are constantly being emitted from us when we speak, eat, etc. And so if a pathogen can hitch a ride on a respiratory droplet, it can become extremely contagious. Okay, so portals of entry. We have the requirement for a pathogen to establish a disease is that it has to get into our bodies. We have some portals of entry of our mucosal tract, and these are going to be relatively simple for the pathogens to find their way into, because our body, with respect to our mucosal tracts, are open to the outside world. So a point of entry can be in your nose, in your mouth. It can be in your genitourinary tract, your gut, and anywhere the pathogen can actually enter the body. There are 3 main categories: our mucous membranes, skin, and parental. Now, just a few things to point out since we have the lungs here. We have our upper respiratory tract. Oops. And this is going to be colonized. And then we have our lower respiratory tract. And this is going to be sterile. So there are some mechanical mechanisms in place that prevent the colonization of your lower respiratory tract, because, of course, oxygen exchange for carbon dioxide would be inhibited if the lungs were colonized, and in some cases, if the mechanical mechanisms were present in the lungs. Now, potential pathogens for this area is going to be staph strep, Haemophilus influenza, as well as Candida. Haemophilus influenza, there is a Hib vaccine for. So Haemophilus influenza was a leading cause of meningitis. It would enter through your lungs and it would get into your brain and cause meningitis, but there is a vaccine for Haemophilus influenza. Now, our mucous membranes, of course, are going to be exposed to the world. We have our GI tract, genital urinary tract, etc. And so there is a lot of surface area for our innate immune response to protect us against. And so there's going to have various mechanisms depending on the site to protect against pathogens. So for the respiratory tract, of course, you have to breathe, and then water is a good mechanism of pathogens to be able to be transmitted because it kind of protects the pathogens from being dried out. It creates moisture. All living things need moisture to some extent. And so it's just a really nice way for pathogens to travel. And so your respiratory tract is not only an entry point, it's also an exit point. Your GI tract pathogens can enter with food and the liquids that we ingest. So in areas where you have cholera, Vibrio cholera, if you don't boil your water properly, you can drink Vibrio cholera and then get cholera. In areas where you have eating meats, if you don't properly prepare certain meats, you can take in bacterium through meats, for example. Your GI tract also has a portal of exit, so a fecal, so there's oftentimes associated, we call it fecal oral contamination. And so people will have, for example, parasitic worms, and they will excrete eggs, and then someone else will come along and touch the eggs and put it in their mouth, or the person will self-infect themselves with eggs over and over again. And so for the upper digestive tract, you have your actinomyces, Tripomona, Neisseria, and Corynebacterium, which can all infect your upper respiratory tract. And then your lower digestive tract are going to be those famous pathogens associated primarily in the United States with food poisonings. We have E. coli or Escherichia coli, hamburgers. We have our Shigella, Salmonella is not listed here, but it is one of them. Candida, which is going to be colonized and can become infectious if you have some sort of compromise. And so the GI tract can be colonized by many different, or it can be infected with many different bacteria. So just a few things about portals of entry or protective barriers for our GI tract and things to keep in mind as we go through this. So the first is you have your stomach acid. And so what's important to understand about stomach acid and pathogens is there are pathogens that have some natural protection from stomach acids, and those are typically the ones that you will find that will pass through your stomach, enter your intestine, lower intestine as well, your colon, and be able to colonize and cause disease. So these pathogens are your gram-negative bacteria and your capsid viruses. So these guys are resistant naturally. And then you have your gram-positive bacteria and your enveloped viruses. And these are susceptible naturally. And so as we go through our pathogens, you're going to see that your gram-negative bacteria, such as E. coli, can get into your small intestine through eating a hamburger and colonize and release toxins and cause intoxication, a colonization. Whereas your gram-positive Staph aureus, if you ingest Staph aureus, you will not get a colonization of Staph aureus in your stomach because Staph aureus gets eliminated by your stomach acid. But what you will get is Staph aureus produces a toxin in the actual food, and you can eat that toxin and get an intoxication from that toxin that was pre-made. And the same with viruses. We have viruses that are just capsids, and capsid viruses are also called naked. And so all viruses have a capsid. So I'm just going to draw a diamond. Here's a capsid. And then you can also have a capsid surrounded by an envelope. And what makes these these viruses different is that on the capsid, you're going to have receptors to infect. And on the envelope, you will have receptors. I should make them different. They're different pathogens. So if the stomach acid breaks down the envelope, which it can't because it's a lipid, so that's what it does. Your stomach acid will break down the envelope as it passes through the stomach. So as both of these pass through stomach, you are going to get your capsid virus intact because it really had nothing to break down. And then you're going to get your envelope virus without its proteins. So this is going to lack infectivity, and this is going to be infectious. So this enveloped virus, which I should have drew as purple, sorry, draw that as purple, just so you can differentiate them. This whole thing is going to be purple. So the purple virus is not able to infect because it lost its membrane, it lost those proteins needed to be infectious. Your capsid virus was not digested or broken down by your stomach acid, so it enters the gut and then it is able to infect. Your genitourinary tract, infections for this are going to be more common in women than men. And that is just due to the anatomical relationship between the anus and the urethra. Contamination, very common in women rather than in men. The urethra in women are shorter, so pathogens can ascend to the bladder much easier. Urinary tract infections can be caused by, for example, Proteus mirabilis, which has urease. And so a lot of the pathogens that are going to be able to infect us have counters to our natural barriers. So urea is very acidic, its natural barrier, and having urease to create sort of a alkaline bubble for your invading pathogens is a virulence factor. So with respect to urinary tract infections, we have a list of potential pathogens. Our E. coli is going to be in both, and E. coli can descend into the kidneys and cause acute kidney failure. We have our Staphylococcus, of course, our normal flora. We have Streptococcus for women. We also have Chlamydia, Neisseria, and Tripomona, our standard sexually transmitted diseases. And then also have Trichomonas vaginalis and Candida yeast infections for women. Now, with respect to the skin is the largest organ of the body. We always have our normal flora. So on our skin is we're going to have staph. Staph are salt-loving. And so they like to be on our skin. Our skin is very salty, and so they are going to be able to colonize our skin very easily. Now, we have two different versions of, or many different versions of staph, but our two main ones are our virulent, which is of course going to be Staph aureus, and our avirulent, so Staph epidermidis, for example. And so your virulent has virulence factors. and your avirulent has no virulence factors. Now keep in mind, avirulent means a normal, healthy individual will be able to clear that infection. It does not mean a person compromised in a hospital situation can. So colonization of catheters by the avirulent staff is a major problem in hospitals. And it's because hospital personnel on their hands have staff. They don't necessarily have to have staph aureus. They can just have any staph. And if they're not careful, if they don't wash their hands, if they don't sanitize their hands, wear gloves when they're putting in a catheter or doing any handling of a catheter, they can add those bacteria to those areas, and then the bacteria can grow and cause an infection. So with respect to skin, of course, the portals of entry are going to be hair follicles, sweat glands, and of course a break in the skin if you get a cut. That's going to make it easy for the bacteria to get in. Now, so other ways to get in is parenteral, so a break in the skin or an injection, or mosquito bites. So this is going to be a break. Injections. That's why it's important to put alcohol, iodine, in an injection site, because the microbes in close proximity to the injection site can gain entry. When you put a small little tiny needle in your skin, the microbes in that area will have access. Also insect bites, so vectors. Our mosquitoes, our bugs, they can carry pathogens and inject them into our skin. So in general, epidermis is no access to blood, and so the wounds are going to be more localized, and they're not going to disseminate. Injected insect bites, where you get deeper, you can have potential injection into blood, and that can of course go systemic. Now staying in, almost all pathogens, if they are virulent, are going to have some mechanism to attach to our cells, our bodies. There's going to be, for bacteria, fimbria, and these are going to be have receptors that can attach to cells in the small intestine, colon, the bladder. This is an example of fimbria. This is going to be E. coli. E. coli uses this to attach to our outer surfaces. There are also adhesions or surface proteins on some bacteria. Neisseria gonorrhea is coated with adhesions and uses also fimbriae. So again, pathogens that have evolved to be able to infect us have these structures to make it easier for them to stay in. Another example is going to be strep mutans. And this is associated with tooth decay. And then we also have, well, let's talk a little bit about strep mutans. Streptococcus mutans is going to have the ability to adhere to the pellicle of our teeth. And once strep mutans binds, it's going to produce a enzyme, a glycosyltransferase, and it's going to break down sucrose. And then sucrose is going to allow other organisms to adhere to strep mutans, and it's going to form a plaque. So plaque in your mouth is basically a colonization of bacteria. And so dentists often have to scrape plaque off teeth. And again, it's because most bacteria cannot bind to the pellicle of your teeth, so they simply get passed on into your digestive tract. But strep mutans can, starts to form a colony, and other different types of bacteria can jump on. So plaque is made up of many different bacterium. Okay, so this is just some terms that you need to understand. So we basically have less virulent, more virulent. So ID, where's ID? Here it is. Infectious dose 50, ID 50, means how many organisms are required to infect 50% of the population. Lethal dose 50 is going to be the number of organisms required to kill 50% of the population. So the organism with the lowest LD50, meaning it requires one organism to infect 50%, ID50, and the lowest LD50 are the most virulent. Okay, so we have the more virulent are the low LD50 and the low ID50, and the less virulent are the high ID50 and the high LD50. And this kind of makes sense. If it takes more organisms to infect a person, they're going to have less virulence. If it's going to take fewer particles of the pathogen to infect us, it's going to be more virulent. On the other side, if it's going to take less of the pathogen to kill us, it's going to be less virulent because you died too quickly to transmit it. If you're going to need more of the pathogen to kill us, then it's going to be more virulent because it has time to transmit. And so you can see on this list here that we have just a range of less virulent to more virulent. So Clostridium difficile is going to be on the less virulent side, whereas you're going to have Francisella tularis or Yersinia pestis plague going to be on the more virulent side. Also, just keep in mind that it's a race between the pathogen and the host response. Pathogens can increase from 1 pathogen to 10 to the 21 pathogen in 24 hours. And so antibodies are very important because antibodies can be produced at a rate of 2,000 molecules per second. So antibodies will have a big role in the initial, if there is a memory response, binding to the pathogens to help get rid of them. So again, it's going to be a balance between the host and the infection and the host's ability to respond to that infection. So again, you have pathogens that have evolved ways to evade our own immune responses. We have co-evolved with these pathogens. There are some passive strategies. This just simply means it's part of the pathogen. They didn't do anything fancy to get it. Capsules are such a thing. Some pathogens have capsules. And this is going to be required for some bacteria to cause disease. If they did not have the capsule, your innate immune response would be able to clear them before they cause disease. So strep pneumonia is such a pathogen, as well as Klebsiella pneumonia. Some are going to have cell wall components that are going to allow it to evade our immune system. We have mycobacterium that have mycolic acid in their cell wall and makes them harder for a macrophage to eat, for example. And then there are active strategies in which there is an active mechanism used by the pathogen to overt our immune system. So passive strategies, again, we have that encapsulation by your capsules, but antibodies can help because they can bind to the capsules and you can get opsonization. So a capsule isn't the only thing needed for a pathogen to get in. It has to have something else as well. We have our bacteria cell walls, and these can protect against pressure. You have Streptococcus pyogenes that has M proteins, and the M proteins are important for adherence. It's one of those sort of adherent proteins that we talked about earlier. M protein also helps to resist heat and acidic environments, so it's also protective. Mycobacterium has mycolic acid in the cell wall. This is very waxy. Um, Mycobacterium also lives within our immune system. So it lives inside of our macrophages, and this protects it against antibiotics. If you have an intracellular pathogen, it's very hard to treat with antibiotics. Antibiotics are very good against our extracellular pathogens. And then we have active strategies. So a lot of pathogens produce enzymes. Enzymes just are simply going to have a function, and the function of these enzymes is to break down things that could counter an infection. So our leukocidins are enzymes made by strep and staph, and they destroy our white blood cells. So they're going to target your leukocytes, your immune system, directly. Hemolysins are going to punch holes into the plasma membrane of host cells, and they're going to, again, lyse your cells. So these are going to be made by staph, for example. Clostridium perfringens, who generates gas gangrene, has hemolysins, and the purpose of Clostridium hemolysins is to generate an anaerobic environment. Does not want to be exposed to blood, does not want to be exposed to oxygen. So it releases hemolysins to break down the tissues in order to sort of wall itself off from the circulation. You have streps that have a streptolysin. Again, it's just going to break down the plasma membranes. We have streptokinase and staphylokinase, which are another series of enzymes that help to digest other cells. We have coagulase, which is an enzyme made by staph infections, and it causes clots to form. And this is similar to what Clostridium was trying to do. Staph is trying to wall itself off and protect itself. Staph can grow in both aerobic and anaerobic environments. And so it's going to create a boil, and then you can't get antibiotics into that boil. You basically have to drain it in order to get the staph infection out. Okay, and that is the end.